In this video, we will be talking about the phototransduction in the rod cell of the retina. This video is for Michael Barton's Wednesday and Friday 11 a.m. class. Phototransduction is the detection of light and then the eye creating a response. So basically what happens is a photon of light is going to go and hit the back of the retina. Alright. And once it hits the back of the retina, it's going to hit two things. It's going to hit the rods and it's going to hit the cones. And these are the receptors that help us to see. Okay, so rods help us to see light, while cones help us to see color. In this video, though, we're just going to be talking about the rods. The rods convert light and energy into neural impulses, which helps the brain to make sense of what it sees. Rod cells consist of three important parts. There's the rhodopsin, which consists of the opsin, and which also consists of the 11-cis retinal. So the opsin and 11-cis retinal together create rhodopsin. Then we have the transducin, and this contains three parts, the alpha, beta, and gamma. And then we have the PDE, also known as the phosphodiesterase. And so basically the goal is to get the phosphodiesterase by itself. As you can see, there's two particles on it. Okay, so step one, a photon of light is going to come and it's going to hit the rhodopsin, which is going to change the 11-cis retinal into all-trans retinal, which makes it a more linear shape. After this, the all-trans retinal is going to unbind from the opsin, which is going to leave the opsin basically by itself. Once the opsin is by itself, it's going to go and it's going to attach itself to the transducin, which is going to activate the transducin, and which is going to make GTP. The alpha subunit becomes active and leaves the other subunits of the transducin behind. Alright, and then the alpha subunit is going to go and it's going to attach itself to the PDE subatomic particle. It's going to take that off and it's going to leave PDE almost by itself. You're going to repeat that step one more time, and then the PDE is finally going to reach its goal of being alone by itself. Once PDE is all alone, it's going to turn cGMP into GMP, which is the important step that leads to our vision. If there is no light, then there causes that to be a lot of cGMP. That means that the cGMP-gated sodium channels are open, allowing sodium to rush in. This causes something known as depolarization. Sodium channels are in constant production due to something called guanylyl cyclase, which is important. If there is no light, then that causes the opposite, which is hyperpolarization due to the sodium channels closed. Light and the reduction of light causes less cGMP. And like I said, this closes the sodium channels. Okay, well that's it. So thank you for watching.